Next, I'd like to introduce Zach Freedman, who's an assistant professor of soil microbiology at UW-Madison. His research focuses on the ecology, evolution, and environmental significance of microorganisms and whether we can lean on microbes to help save the world. So welcome, Zach.
Thank you, Mary.
All right. So yeah, my name's Zach Freedman. I'm an assistant professor, soil microbiology. Thank you, Mary, for the invitation and for bringing us all together today. So for the next 20 minutes or so, I'm going to talk to you all about the importance of microbial communities and their ecosystem functions in bioenergy systems. And I would like to first start with some acknowledgments of some folks' work that I'm going to be highlighting today. Before starting here at UW-Madison, I was faculty at West Virginia University and very active in bioenergy research out there. Jenny Kane was a PhD student on the project I'll highlight. She's now a postdoc on the same project, which is great. And the co-PIs on the project, Dr. Ember Morrissey, Lewis McDonald, and Jeff Skousen, and a whole team of undergrads that made it happen. So, microbes are abundant in soil and do lots of good things, great things, I will argue. Microbes certainly are also abundant on plants and in plants, but given it's World Soils Day today, happy World Soils Day to everybody, I'm going to just totally ignore the microbes in and on plants and focus on the microbes in soil. So first, microbes in soil, they are abundant. Okay, if you look there between bacteria, actinomycetes, and fungi, highly abundant in soil and also constitute a high amount of biomass. We can thank microbes for lots of things. Just to calibrate people to their importance in the room, one, they are imperative in decomposition of organic matter. Just we get lots of organic matter building up, if not for the microbes. Microbes build soil structure that contribute to soil aggregation, for example, as well as build stable soil carbon. We can thank microbes for the mineralization of most major nutrients required for plant productivity, like nitrogen and phosphorus, this owing to their power in decomposition. Microbes can also fix atmospheric CO2, so act as a natural fertilizer, fix atmospheric nitrogen. So act as a natural fertilizer, bringing in nitrogen gas from the atmosphere, fixing it then into forms that are usable by plants and other living things in soil. Microbes can engage in myriad of beneficial plant-microbe interactions. So think mycorrhizal fungi that can act as an extended root system of plants. And also, a healthy microbial community can improve the resilience of an ecosystem under different pressures that it may face due to climate change, extreme weather events, etc. And I just want to give a shout out, I see Randy over there, and Randy and I like to argue about whether plants or microbes are more important to a system. I just want to put out there that, of course, plants can benefit a microbe in giving microbes a nice home in order for them to do their thing. So. But really, microbes are in control. They'll be around a lot longer than plants will on the planet.
The plants are just conduit of carbon for the microbes, right?
Exactly. Now we're talking. And one ecosystem process that I'm very interested in is carbon sequestration, the movement of carbon. This is an emergent property of soil. Lots of things have to happen in order for soil to build or lose carbon. And it is very well established at this point that the inclusion of microbial data improve predictions of future soil carbon. And that's shown by these figures from a paper by Will Weider and others. On the top map, that shows the observed distribution of soil carbon in the top meter of soil. On the bottom left, this is a model example that does not include microbial data. And then in the bottom right are model results that include microbial data. And basically here, the map on the bottom right looks more like the map on top than the map on the bottom left. And so the take-home message here is that incorporating, again, microbial data into these Earth system models improve predictions of carbon in the future. So this begs the question, can we, given microbes are imperative to the trajectory of soil carbon, can we manage soil microbes either directly or indirectly to improve not just soil carbon, but also these vast ecosystem services that microbes just do? So with this COVID era of review paper writing, these two very similar review papers came out at around the same time that has very similar questions. How can we best either directly or indirectly manage the soil microbiome to boost soil processes? And the take-home messages of these two were very, very similar, and also similar to some of the themes that have been coming out from the late morning talks here. But one is that context is hugely important, right? That the relationship between soil biodiversity, soil carbon, and plant yield depends on the place. It depends on the place and time. And two, that degraded lands are especially promising candidates for the management of soil biodiversity for a sustainability benefit. Okay, so I'm going to argue here for place-based thinking in terms of applying these management strategies for soil to maximize soil microbial benefit in the context of bioenergy systems. And so I'm going to highlight some work from my previous position at West Virginia University, where I think we did a really great job, if I do say so myself, in having a place-based approach to using bioenergy systems to maximize the sustainability overall, that is environmental sustainability, human sustainability, all the three pillars of sustainability I think we target here, where we're trying to repurpose land that was used for surface mining activities to grow bioenergy crops. Okay, so it's place-based in terms of trying to keep energy land in energy, and using these beneficial plant-soil microbe interactions to build more functional soil. And while the region that I'm going to focus on here is the Mid-Atlantic and Appalachian region, I think a lot of the same thinking and the same kind of project structure could work in terms of trying to maximize the use of marginal lands in the Midwest region as well.
Is it, say, Wyoming? Is that a typo there?
No. So these are two examples of giant surface mines.
Okay.
One is in West Virginia, the Hobet mine, that was at one time the largest mine east of the Mississippi. And then next to it is the Black Thunder mine that currently is the #2 producing coal mine in the country. It just looks much more impressive than the #1 producing coal mine in the country, which is just up the road in Wyoming. So just showing these as an illustration of the destruction that surface mining can cause. So again, owing to the place-based effort here, for this system we chose giant grass Miscanthus giganteus as our model bioenergy crop. Why? Is because modeling efforts, like you see the results here from Davis and others, have shown that Miscanthus is great at building soil carbon. Okay, the plant has very high nutrient use efficiency, high water use efficiency, and a robust root system that we think would serve it quite well in these highly degraded post-mining soils. And I just want to be clear, certainly I'm not trying to argue here that Miscanthus is this magic plant that we should grow everywhere. Just saying for this place at this time, it was what we thought was the best of the options. So we asked the general question, does Miscanthus production alter plant-soil microbe interactions to ultimately increase soil carbon? And from this cartoon here, kind of illustrates the dynamics that we were interested in, that my lab is currently interested in now, whereby the plant fixes CO2, kind of brings it down into the soil through rhizodeposition or litter fall as plant carbon inputs. And then, let's see, can you see the, no. And then we see the soil microbial community in the brown circle there, and the microbial community processes this carbon, and it can eventually end up either going back up into the atmosphere as CO2 in a process we call respiration, being assimilated into microbial biomass, and that's one way of carbon staying in the soil. And also this microbial processing can ultimately result in stable soil carbon. And we're particularly interested in this side of the model, the microbial role in this. One particular parameter that we're interested in is called carbon use efficiency. And microbial carbon use efficiency, it's the ratio of carbon allocated to new microbial biomass over the amount of carbon consumed. And the amount of carbon consumed is the addition of carbon that is respired plus the amount of carbon allocated to biomass. Why you think this parameter is of interest. One is that it's directly incorporated into Earth system models or predictive models to predict the fate of carbon in the future. And it's I shouldn't say it's easy to predict in the lab, but it's not terribly complicated either. And also, from results of a meta-analysis, microbial CUE has been shown to correlate with soil organic carbon at the global scale. Okay, so it's a pretty powerful parameter in that way. And I'm just we had hypotheses, it's good science, I'm just gonna skip over it in the interest of time. So for this study, we selected 3 locations that differ in land use history. We had a low disturbance site that was unimpacted by mining, a moderate disturbance site that was adjacent to surface and underground mining activity, and it had altered hydrology and soil properties. And then we had our highest disturbance site that was directly surface mined and unreclaimed. So there were chunks of coal in this soil that we discovered as we were digging. So while I guess it wouldn't fall under the definition of biochar, I like to think of it as like the original biochar, like OG kind of biochar, which makes it kind of an interesting system. And I know Thea can forget I said that, please. So first, just to establish some data that we found in the first 3 years of the study, let me calibrate you all to some colors that I'm going to be showing. Most of the graphs I'm going to show you will have year on the x-axis and the different levels of disturbance as indicated by the different colors here. So over the first 3 years of the study, the plant biomass increased the least in the highest disturbance site, which is not surprising. And also we see soil carbon on the right that increased over the years, but not as much as we expected, except for in the moderate disturbance site. And I just want to point out too, the amount of soil carbon in the highest disturbance site was surprisingly high. What we think is going on here is it's reflective of the coal that's in the soil. So it's a high carbon soil, but that carbon isn't necessarily kind of usable by organisms that need carbon at the rate at which they need it. So we measured microbial biomass carbon. As expected, it increased over time. Microbial biomass carbon is another one of these parameters that is directly used in predictive models of soil carbon. So this was expected and good to see. We looked at particular microbial groups that are shown to be of benefit to plants. So one of which is arbuscular mycorrhizal fungi. They act as an extended root system, if you will, of these plants. Here we have results of correlations over the years of the different disturbance levels, and found that, perhaps unsurprisingly, but interesting, we found it in the highest disturbance site, the most AMF were recruited over the 3 years of this study, which makes sense. Those plants would be most in need of the boost that AMF can provide. And we also see the slope of the line in the moderate disturbance site. It's about to overtake the AMF in the lowest disturbance site. So these plants are recruiting beneficial microbiome in the first 3 years of stand establishment. We also see some change in the richness, so the number of species that are present. So bacterial richness increased over time, and also bacterial evenness increased over time. This is how kind of similar in number different species in the community are. So the community is getting more kind of evenly distributed in terms of its species abundance. And the slope of that line is most extreme in the highest disturbance site, which is pretty cool. So we see the most change over time in the highest disturbance site. This is a busy graph, so allow me to walk you through it. This is showing that Miscanthus production is selecting for particular members of both the bacterial and fungal community. So we have bacterial data on the left, fungal data on the right. And the more blue those boxes get, show that Miscanthus is selecting for a greater abundance of these particular microbes over the business as usual soil sample that we took, which was mixed cool season grasses. I wouldn't go so far as to call it a prairie, but the closest you get to a prairie in the mid-Atlantic region. We put these boxes around the different microbes to emphasize saprotrophs, who are microbes that are they decompose organic matter, and decomposition is generally important to the functioning of soil. And also in the brown boxes, we have known plant growth promoters. So again, Miscanthus, this giant grass, is recruiting these beneficial microbes to its roots, which will benefit the plant and also benefit the soil in which it grows. So now let's get more into function. So this is carbon use efficiency data over the first 3 years of the study. See that carbon use efficiency increases over time, which is kind of good for the trajectory of soil carbon. And we find that carbon use efficiency is lowest in the moderate and high disturbance site. This is likely owing to microbes having to spend more energy in maybe stress response activity or general maintenance activity, that they're just, you could think of them as not being as happy as the microbes that are in the least disturbance site. And like has been shown in that meta-analysis, that carbon use efficiency tends to correlate with soil carbon. We wanted to see if that held true in these unique sites. So we did just that. You see the results of the correlation in the different colors on the top right there, and found that indeed in this system, soil carbon correlated with carbon use efficiency. That correlation was strongest in the moderate disturbance site and lowest in the lowest disturbance site. We then aimed to determine whether carbon use efficiency was related to these microbial parameters. So we regressed carbon use efficiency against both bacterial and fungal diversity. And we found that microbial CUE strongly correlated with bacterial diversity at all sites and did not correlate at all with fungal diversity across these sites. And the correlation here was strongest in the moderate disturbance site, lowest in the highest disturbance site. So here there seems to be some correlation, not a direct causative link. We're just not able to show that with these data, but a correlation anyway between microbial parameters and carbon use efficiency. And last, and I think what is the coolest part of this study, is that we found that Miscanthus production homogenizes soil parameters across disturbance levels over time. Okay, so allow me to calibrate you all to this figure. What you're looking at here is called an ordination, where dots represent a particular experimental plot. And if two dots are closer together, it means that those plots are more similar in the parameters that we threw into this ordination than dots that are further apart in this ordination space. And how we produce this ordination is that we combined all of the different soil data that we generated there that you can see on the far right, calculated a distance matrix, and then the product of this distance matrix is visualized in this plot here. The different shapes indicate the year of study. So where you see circles, these are data from 2019. If you notice there, circles tend to be over on the left side of this figure. Okay, triangles represent 2020, the second year, and the triangles tend to be towards the middle of the plot, and then the squares are 2021, they tend to be on the right of the plot. So as you go from left to right, this is representative of time. Then if you look at the spread of the points, over on the left, they're the most spread, and then as we go to the right, they tend to come closer together, okay? So, between the low and the medium and high, we see a homogenization or an increase in similarity of the soil properties across the 3 years of Miscanthus growth. So ultimately, this highest disturbance soil is looking like, microbially, soil biologically speaking, is looking like the least disturbance site only after 3 years of Miscanthus growth. Okay, which is pretty cool. So this is some evidence that Miscanthus is kind of healing this highly disturbed land to make it more like the least disturbed land. So if you allow me to kind of bring it all together here. So the story I think we're trying to tell, that I hope you kind of buy after this, is that Miscanthus growth increases microbial biomass carbon, increases bacterial diversity, did not have huge impacts on fungal diversity or evenness. Then these increases in bacterial diversity and microbial biomass were associated with increases in microbial carbon use efficiency. And microbial biomass carbon was also associated with increased microbial carbon use efficiency. So the microbial cycling of carbon was occurring in such a way to keep that carbon in soil rather than it being blown off into the atmosphere as CO2. And then, lastly, as others have shown in meta-analyses, and we're showing here that the increases in carbon use efficiency correspond to increases in soil carbon and soil organic matter. So ultimately, Miscanthus seems to be doing what we were hoping it would do in the system by leveraging the soil microbiome to help enhance the carbon sequestration in soil. So to kind of wrap up this case study, we found that Miscanthus production facilitated the improvement of soil properties across disturbance levels. Something that I did not share, we had this whole amendments side to this study. None of them had any impact on anything, which was, a bummer, but the results were very clear that amendments didn't influence these trends, and ultimately Miscanthus production homogenized soil properties across disturbance levels. I was going to bring it back to the hypotheses, but I'm going to, instead end on a bigger picture. I'm going to wing it a little bit here. Like, what's the take-home message about microbes and their role in, sustainability and life cycles? That I think, building upon what Aaron and Randy and Thea were starting to build on, that, place matters, context matters, and that's true for microbes also, right? And so I think it's important to use a place-based type of plan to try and best leverage soil microbes to enhance the overall sustainability of a system. And also, given, learning about life cycle analyses and talking about them more, to, generate microbial data that isn't necessarily about how does the plant growth affect the microdiversity of a certain, species of bacteria that I may be super interested in, but modelers couldn't care less about. So to work closely with modelers, right, to produce microbial data that can inform and improve those models, and to talk more with modelers, think there may be some microbial data that they may be overlooking. So you may want to think about how to improve Earth system models such that we can best predict soil carbon moving forward into the future. So with that, thank you all, and look forward to, we can talk later about any questions that you may have.
Thank you, Zach.